eye just as it continues in a normal circle, but it actually protrudes forward. And this is clear. So I challenge you to find somebody, an adult or a peer or a small child. And just pinkish is because this part right here below the sclera is known as the choroid. And the choroid is a thinner area, but it's covered in blood vessels, which makes it pink in real life as well. As you come forward, you get to the part of your eye. And the lens sits right behind the pupil and the iris. And the lens's job is to focus that light that's coming in through the pupil onto the retina. And so the retina is the deepest of the three layers of the eye. And so this is where the photoreceptors are going to be located. And there also is the deepest layer, which I'll show you on a slide later on, which is a pigmented layer that absorbs whatever stray lights that are not going to be picked up by the photoreceptors. And by doing this, this helps with visual acuity. So it's kind of like when you're trying to look at something like here and you block all the light at the side, so it helps you focus on what's right in front of you. So there's something else that's actually on the surface of your eye called the conjunctiva. And this is a thin epithelial layer that covers not only the white of your eye, the sclera of your eye. You can see it right here because they cut it to show that it's covering the surface there, but it also covers the inside of your eyelids. And it contains a lot of blood vessels to help nourish your eyelid and your cornea and help keep your eye from drying out. Now, when people become aware of the conjunctiva is when you have something called conjunctivitis, which people commonly call by the slang term pink eye. Okay? So conjunctivitis is itis, inflammation of the conjunctiva. And this can be due to allergies, very common due to allergies, very common in the summer due to chemicals in the pool. If it's due to an infection, it can be due to bacterial or viral infection. The most common infectious cause is viral infections. If it's due to a virus, they're not going to let you into the community pool. But a lot of people get conjunctivitis from playing in the pool. So if you know you get conjunctivitis from the pool, don't let the lifeguard keep you from going into the pool if your conjunctivitis is only from going into the pool. All right, so let's go back and talk about that lens. So when light is entering the eye, it hits the cornea first, which bends it minimally, but it does bend it a little bit. And then it travels through the liquid, which is in the eye, until it hits the lens. And now the lens is curved. And so its curved shape is going to bend the light rays, just like when light hits a prism, it's going to bend it. And the whole point of bending the light rays is that it will focus it on the photoreceptors on the retina. And when you are looking near versus far, the shape of the lens is going to change so that it will still focus on the retina. So here's how that happens. You know, I talked about we had the ciliary body, so we had a muscle here, and then we had ligaments which were attached to the lens. So when these ligaments are pulled tight, the lens is pulled, and so it becomes very thin. The diameter gets big, and the lens becomes very thin. And so this is what happens when we are looking at a distance more than 20 feet away. So this essentially is the resting position of the lens. It's how your lens is most of the time. But when you want to look at things that are closer than 20 feet, like you want to look at your computer, when you want to read, when you want to eat, then we have to change the shape of the lens. And so what's going to happen is that muscle is going to change, and thereby changing the ligaments, and the ligaments are going to become very lax. And that lens, instead of having a big diameter, is going to have a small diameter, and the lens is going to bulge and become very fat. And so it's going to change the focal point. And so this also has to do with whether or not you have the blurry vision associated with nearsighted and farsightedness. And I explain this because I know my middle child did not understand exactly what was happening in this when he was in college. If you are nearsighted, it means you can see things that are close to you, that are near. And things that are far away are blurry, which means without glasses, you should be sitting in the front row of the classroom and not in the back of the classroom. In the movie theater, you should be sitting near the front. And here's the reason why you are nearsighted. It is because when it's being focused, either the lens is too strong or the cornea is too thick, not that common, or your eyeball is just too long. And so it is focusing in the middle of your eyeball instead of on your retina. But this is very easy to fix. We just put a lens in front of your cornea that is biconcave, and so it makes the focal point longer so that it will focus on your retina now. And now you will have normal vision. Now, if you are farsighted, it means you see things that are far away clearly, and trying to read things up close, they are very blurry. So it means to see clearly in a classroom, you would sit in the back row. And that's going to be because your lens is weak or your eyeball is too short. So maybe as you grow and your eyeball gets bigger, you will lose your farsightedness. So maybe you would only need glasses as a child until you hit your 40s, and then you need glasses later on. But we couldn't put the same kind of lens. We would have to put a convex lens to shorten the focal point, to bring it to your retina. And this is great until, as I said, you hit your 40s. And by the time you're 40s, your lens is tired of going fat to skinny, fat to skinny, fat to skinny, and it just gets tired and it just doesn't adjust so much. And so that's when we start needing reading glasses for up close. And nowadays, because we have computers, reading is about 12 inches away and computers are 18 to 24 inches away, that we have regular vision for distance, and then we have one set of corrective lenses for computer and a different set of corrective lenses for reading, which is why I often have my lenses for computer when I am giving these talks, but I actually can see okay on my computer. All right. So let's get back to the eyeball, because in the eyeball, we have spaces that are filled with fluids. So we have to learn the names of the spaces and the fluids as well. So behind the lens and the muscle and the ligaments, we have this big space known as the vitreous chamber, and it is filled with a jelly-like substance. So think of a jello jiggler known as the vitreous humor. So the vitreous humor is in the vitreous chamber. And this, because it's like a jello jiggler, is giving a pressure. This is what keeps your eyeball shape. Otherwise, it would collapse down on itself. This is formed before you are born. It is formed once in your life and it's never replaced. So if you had a knife to your eyeball and it leaked out, it would be gone. And doctors would have to put fake vitreous humor into your eyeball. If you ever have floaters in your eyeball, it's because of crystals and proteins in your vitreous humor. And what happens over a couple of days, they end up settling at the bottom and the floaters go away. Now, in front of your eyeball, you actually have two spaces connected with the pupil in between them. So between the cornea and the iris, this is the anterior chamber. And then between the iris and that suspensory ligaments and ciliary muscle, that would be the posterior chamber. So the anterior and posterior chambers, because they're connecting, they're filled with the same fluid, which is called the aqueous humor. And it's called the aqueous humor because aqueous means watery. And this is a water-based fluid. And in fact, your body replaces this about every hour and a half. So it's mostly water. If you have a high blood sugar, it'll have a higher content of sugar in it. And the whole point of this fluid is to nourish the inside of the cornea and to nourish the lens. Remember that conjunctiva is nourishing the outside of the cornea. Now, this aqueous humor and the anterior and posterior chambers is very much related to glaucoma. So let's look at glaucoma. So here is the sclera and here was the cornea. So between the iris and the cornea, this was the anterior chamber filled with the aqueous humor. This was the ciliary body. There were the ligaments, and then here is the lens. So this is the posterior chamber right in here. Now, this aqueous humor is actually made right here, in the ciliary body. It gets released into the posterior chamber, then travels through the pupil into the anterior chamber. And then we'll go back into the circulation through these little openings known as the canal phlegm. So on this other picture, it's going to leave the blood, the watery substance is going to leave the blood, go here. Whoops, where's my arrow? It's going to leave the blood here, go into the posterior chamber, bathe the lens, go through the pupil into the anterior chamber, bathe the front side of the iris, bathe the inside of the cornea, and then go back into the bloodstream here in the cornea. So in the corners, sorry. Now, what happens if you either make too much of this aqueous humor or if these little canals are blocked and you cannot reabsorb it? Well, then you would have too much aqueous humor, which would mean you would have this part would enlarge and you would have too much pressure there. Okay. And then what's going to happen is this is going to be pushing backwards here where that vitreous humor was in the vitreous chamber. It's going to be pushing everything towards the back of your eye, towards where the retina was on the choroid. It's going to be pushing on the blood vessels in the choroid. It's going to be pushing on the retina. It's going to be pushing here where your optic nerve is trying to leave. And so the cells of your retina are going to start dying. Your optic nerve can be damaged and you can become impaired. And this now is like the #1 cause of preventable blindness here in the United States. So definitely go get your eyes checked. Have you sit on a little machine, rest your chin and get a little puff of gas and then measure the pressures in your eyeballs. Not that expensive to get your eyes checked every year. If you don't have eye insurance, go to any Costco or Sam's Club. You do not need to be a member to get your eyes checked there. It's pretty cheap to get it done. All right. So let's talk about what's going on in your retina. In your retina, we've got multiple neurons. The ones you know about are your photoreceptors, your rods and your cones. Here's what you look like. Look how tiny the rods are the cones are compared to the big rods. I know the diagram makes them look the same size. They are not. Those cones are tiny compared to the big rods. But when you look at them, so at this end, there are all these little discs of pigment. Kind of makes you think of what's going on in trees, photosynthesis, and all those pigments there. Okay, and all those discs. Okay, so these are modified neurons. Okay, where these are the receptor parts, and these are the axon terminal parts. Yes, kind of weird. And so in these pigment discs, there are molecules which are derivatives of vitamin A. So think of like vitamin A as found in carrots. Okay, so it's related to the carotene that you find in carrots. All right, so if you don't like carrots and other orange vegetables, you're going to be deficient in this pigment. You're going to be losing your vision. Okay, so the rods become activated in low light, but they only give you blacks, whites, and grays. And the rods are in the periphery of your retina. Whereas your cones, and most of us are 3 colors, not your 3 primary colors, but blue, green, and red activate them. And they're more in the center of our retina. It takes more light to activate them. So if you're walking into a dark movie theater, you're not activating your cones. It's going to take 30 seconds for the cones to start being activated. Okay, so that's why, like at nights, if you car comes rushing down and hits other parked cars, you can't tell if that car was dark green or dark red or dark blue. Like all of you are going to say, was it black? Was it gray? Was it green? You don't know because only your rods were being activated. Okay, so here's the rod and cone placement. If you look in the periphery, all rods and only near the center is cones. And you see this area here that's white? That's where all the axons are exiting your retina. So that's your blind spot where your optic nerve is going to be. You have no vision there. Do you notice that's not right in the center? That's off to the side. Because if that was right in the center, when you look straight ahead, you would be blind. So it's off to the side on the right and to the left on your 2 eyeballs, which is really good. All right. So when we look in the retina, we actually are going to have 3 layers of neurons we're going to do. And so when light comes in, the rods and cones are not the first layer. It has to go through all these other layers and go all the way to the deep part to find the rods and cones to be activating. And here, right here, these are those pigmented cells that I talked about previously to block all the peripheral light that's not going to be activating the rods and the cones. And so once the rods and cones are activated, there's going to be a series of reactions that are going to occur. And then the messages are going to cause a bipolar cell to send a transmitter to a ganglion cell, which is going to be activated, woo, action potential to send a nerve impulse. Okay, so this is kind of backwards. We have to go past the ganglion cells and past the bipolar cells to get to the rods and cells. So I don't want you to say retina anymore. Now you need to say rods and cones to the bipolar cells, to the ganglion cells. So now we finally have an action potential. Okay, because these three cells need to synapse with each other. Okay, so light is splitting the molecules in the photoreceptors, causing a whole bunch of reactions, causing the bipolar cell to release the neurotransmitter, causing the ganglion cell to have the action potential, which travels down the optic nerve, which exits the back of the eyeball. Okay, but that doesn't give us vision, because by now we know to have vision, we got to get it all the way to the cortex. So how are we going to get there? And are we going to involve the sensory relay center? Well, let's go to another slide to find out. But before we get there, let's look at a couple special areas of the retina I want to draw your attention to. If we go straight back through the pupil, straight back to the middle of the eyeball, do you see this little pit-like depression called the fovea? Right in the center of the eyeball. This is the fovea, where we are loaded with cones and very few cells. This is our area of sharpest vision, which is great. You want to see sharp, look straight ahead. You want to just get an impression and just recognize movement and not be able to tell colors, go to the periphery. And then this part right here, which is just axons exiting, this is your blind spot. It's where the axons of those ganglion cells are exiting, and as soon as they are outside the eyeball, we call it the optic nerve. There are no photoreceptors here, meaning there is no vision there. Okay, so now here's the optic nerve exiting one eyeball. Those axons are in blue. Here's the optic nerve exiting the other eyeball. Those axons are in yellow. Look what happens. The axons coming from the middle side of each eyeball cross. So the ones from the right side of your eye are not going to the right side of your brain. The ones from the medial aspect are going to the left side of the brain. The ones from the lateral aspect are staying on the right side. And the same thing for the other eyeball. Yes. So that means if you wipe out one side of your brain, you have not wiped out all the vision from that eyeball. See, brilliant. Okay. So after we've crossed, we are now inside the eyeball. So now we have axons inside the eyeball. So we call it a tract. Because remember, a nerve is axons outside in the peripheral nervous system. A tract is axons inside the central nervous system. So this tract is now the axons inside the central nervous system. So where are these axons going to go? To our sensory relay center. Yes, they go to the thalamus. Now, why is it showing few neurons going to other places? Because, for instance, if you're watching people playing tennis, then you would have to send stuff elsewhere. But we don't care about that for this course. All right, we're going to go to the thalamus, and from there, we are going to go all the way to the back of the brain, because you know by now that the occipital lobe is where the visual cortex, the vision part of your brain is. And then for us to be aware of whatever we saw, we have to take it all the way to the gray matter, to the visual cortex. But did you know it's even more complicated? There's two more complications that occur. The first thing is that because we have two eyeballs in front, as opposed to other animals which have eyeballs on the side, which give independent information, because our eyeballs are in the front, our visual fields cross. You see this green area where our visual fields cross over? That gives us depth perception. That's really kind of cool. If you only look with one eye, it's really hard to tell depth perception. But with two eyes, it's very easy to see depth perception. All right. The second thing is when these messages are sent to your brain, they are actually sent upside down and reversed. And your brain figures it out. So when you were first born, it takes you a couple of days to figure this out. And we know this because we can give people glasses that will do the inverted and reversing because now your brain is so we know that our brains have it inverted and reversed. So we can give you glasses that will invert it and reverse it again so that you will see it inverted and reversed. And so then when you start walking, like you're very reaching for the wrong side and you think you're walking up when you're walking down. And that literally takes you about 24, 48 hours, after which, even wearing those glasses, you're totally back to normal. So it's really kind of cool. Your brain learns how to cope with it. And so that's it with vision. So come on back for the next thing where we're going to learn all about hearing. See you soon.